So our E. coli are these right here, and they have a flagella to move, which is seen with a pipe cleaner. And so E. coli are waterborne diseases that come from, in this case, contaminated water. So our cow has feces that contaminate the water that's used to clean up our produce, which is lettuce in this case, that we eat. And because we digest it, the skin doesn't really act as a barrier defense in this case, but our mucus does. So it stops some of the E. coli that enters. But the E. coli that gets past this defense goes into our intestines and other digestive systems. So over here in our intestines, there are macrophages that act as like another level of protection. And what they do is that with the TLRs, they detect the E. coli and then they engulf it and eat the E. coli. And then from there, we also have neutrophils. And so these are white blood cells that also eat E. coli. There are both free and fixed types of macrophages, where the free types roam around on tissue and eat the E. coli, while the fixed types are attached to fibers and eat E. coli that way. Okay. And so what the neutrophil do is that they also engulf E. coli or and other pathogens like macrophages, but once they engulf one of them, they die. And also, some of the white blood cells secrete antimicrobial proteins, which are seen with these, and some of them attach to the E. coli, stopping cell-to-cell spreading, or they also attach to macrophages that activate the macrophages to eat the pathogens. And going down, the inflammatory response is what happens when the E. coli enters it. It leads to swelling in the intestine. And what happens is that cytokines and other chemicals act and like signal the intestine to swell up. And that leads to some more macrophages and neutrophils entering the intestines to eat up the E. coli. However, another layer is that in case the E. coli actually do infect a cell, which is right here, in case they do infect the cell by passing on their protein and other chemicals onto the cell, which makes them become infected, that means that they don't secrete a protein called MHC1. And when they don't secrete that, natural killer cells know that, and they attack these cells and cause apoptosis. And these natural killer cells are usually found in the epithelium.
If the E. coli bacteria get past the natural killer cells and the other barriers present within the innate immune response, then the bacteria infects the cells, causing the primary adaptive immune response to occur. In a cell-mediated response specifically, the infected cells present antigens in red to T cells using the MHC complex. Antigens are then presented on MHC1 of the infected cell in pink, which the cytotoxic T cells then recognize and bind within the epitope binding site to the MHC1 of the infected cell with the help of CD8, a surface protein. This binding activates the cytotoxic T cell. This makes the cytotoxic T cell an active killer, meaning that it secretes proteins, proteolytic enzymes, and perforins that then destroy the infected cell along with the E. coli of the infected cell, which is shown here. The engulfed materials then proceed along to the macrophages. If E. coli infects a cell and engulfed materials are present within macrophages near B cells or dendritic cells, then the cells will present antigens, which are then shown in red, on the class 2 MHC complex, shown in purple, from the engulfed materials. Helper T cells recognize these specific antigens and bind within the epitope binding site to the MHC2 of the infected cell with the help of CD4, another surface protein. And this binding activates the helper T cell. The activated helper T cell secretes cytokines that strengthen the immune response by stimulating macrophages, lymphocytes, and then B cells to make more antibodies. For example, the macrophages here will then destroy more engulfed material, such as this destroyed E. coli. In the case of the humoral response, an antigen present from the E. coli infection, which is then shown in red, will bind to the B cell, having a receptor specific only to that antigen. The selected B cell then proliferates, forming a clone of identical cells bearing a receptor for the antigen. Some of these daughter cells developed into short, lived plasma cells that secrete antibodies specific for that antigen. And then this will help destroy the E. coli, which is shown over there.
Here we have the E. coli. It's with the memory cells, and memory cells are part of the adaptive immunity system and will form the secondary immune response. And what we want is that the secondary immune response will make it so that the next time an E. coli enters the body, both the T memory cells and the B plasma memory cells will allow the process to start again. First, it will signal the cytotoxic T cells to destroy the virally infected cells by the E. coli as the antigen is presented on the MHC class I molecule. And by doing this, it will allow for these virally infected cells to die off again. It will also be put within the B lymphocyte in order for the same antibodies produced for the first response to come back and be made more into much stronger and quicker response and allow for the body to react into the future as well. If our bodies are not able to produce the antibodies that we saw within our adaptive immunity system to E. coli, and they are challenged by E. coli, one temporary solution is passive immunity, in which, as we see here, our human is not able to produce the antibodies needed to attack the E. coli. So, what do we want to do? What we want to do is to inject the human with a shot of antibodies from a cat, which is immune to the E. coli, and produce these antibodies, allowing for the human to attack the E. coli. However, this is only temporary and lasts a very short time, in which it could die off after a couple of weeks, as these antibodies are very short-lived and will not be able to recur within the body. The reason we give this example is because recent studies have shown that in mice, passive immunity can be passed on to fight E. coli through the mother with OMV shots or outer membrane vesicles. And this can be even applied to future human studies one day, in which some people are not able to produce the correct immune response to E. coli.